So this is Dr. Poe, and today I'm talking about the Bios242, week 2, just an overview here. So this week we're really talking about the nitty-gritty details of bacteria, archaea, eukaryotes, and viruses. So in viruses, I talk about those in a different video, so I'm not going to talk about this. For the eukaryotes, the main groups, of course, Plantae and Animalia are not really discussed in microbiology classes. Animalia. With the exception for the animals, we've got some arthropods that are important. Arthropods. So, like ticks, and some insects like mosquitoes that carry diseases. We also talk about the helminths, the worms, so like tapeworms. So you should, be familiar with their roles. These guys are vectors of disease. That means that they transfer diseases. And helminths, actually, those are, intestinal parasites, so those are important. Plantae, we kind of talk about algae for a second. So really the important role, there's some microscopic algae, but algae's real main contribution is that it produces the agar, to make our Petri plates. And remember that agar is something that most microorganisms cannot eat because it's so amazingly complex, really complex carbohydrate there. So this is not a carbon source for our bacteria. When we're growing them, it just gives us a solid surface to actually look at colonies. Okay, so bacteria and archaea, those are probably ones that you haven't thought too much about. So archaea, this week we're going to learn that there may, we learned last week that these are both our prokaryotes, they're single-celled, they're small, they do not have any organelles. They don't have any membrane-bound organelles. I know some textbooks call ribosomes organelles. Ribosomes are not organelles. They don't have a membrane. So these guys are small. They do have ribosomes, but they don't have organelles. All their chemistry takes place in the cytoplasm. Their DNA is stored in the cytoplasm in a dense space called a nucleoid. But anyway, so this week we're talking about their differences. So archaea, these are typically extremophiles. That's where they live. Extreme environments. So they love high salt, and that can make them halophiles. That's the word if you live in high salt. They live in deep pressure, and so that makes them barophiles, some of them. They can live in high temperatures, like at Yosemite. So that makes them thermophiles or hyperthermophiles. Hyperthermophiles if it's like basically boiling water. And as you can imagine, these are not the environment of our body, so that's why they don't grow well in our body, because it's outside of their specific range. Our body likes more mesophiles that are middle ground, middle pH, middle salt, those types of things. And so these extreme environments, there's no examples of any pathogens from archaea. Some interesting groups of archaea include the methanogens. Those are really interesting because they fix carbon. They can take CO2 from the atmosphere and make CH4, and so that's a reduction. It's really high energy to do that, and it's absolutely fascinating. And maybe we can use these as an alternative fuel source, and that's really cool. But they're not going to cause any sort of diseases. So they are interesting to study, but not necessarily for other reasons. Not for pathogenic reasons. And then, of course, our bacteria. These live in all types of environments. Some of them, there's not an environment they don't live in. So there's some pathogens, there's mesophiles, there's extremophiles. They have all different types. These also have different shapes. And the cell shapes we call morphology. Absolutely know that word. Morphology. That means the cell shape. So are they circle-shaped or sphere-shaped? Then we call it a coccus. If it's a rod shape, we call it a bacillus. And if it's a spiral shape, if it's a flexible spiral, we call it a spirochete. If it's a stiff spiral, we call it a sporillum. It's hard for me to draw the difference between those two. It's also possible for it to kind of have like these branchy filament things. So we call those filamentous. And it's also possible to have like a change of shape or not a defined shape, and we call that pleomorphic. So remember, morph means shape. If you're morphing, you're changing shape. Morphology just means the shape that it takes. Okay, when these undergo cell division, they have different arrangements, and that's how cells are attached after cell division. And so arrangements can be like in a chain, like strepto. This example is a chain of spheres, so we would call it streptococcus. We can also have a chain of bacillus. We would call that streptobacillus. They can be in grape-like clusters, which I'm sure you've heard of this one. It's called staphylo, like a staph infection, because that's what it looks like under a microscope. Bacillus typically don't do this. We can have just two spheres, two rods, and we call that diplo, di meaning two. What else? What am I missing? Oh, we can have a group of four. We call that a tetrad. We can have like a cube type shape. So if you can imagine like this tetrad then divided again to make like 8. I'm really bad at drawing this, guys. That's sarcinae. And then the last one that I want to mention is like the fence posts, which typically bacillus do. We call that palisades. Okay, and so you've noticed some of these words maybe before, and it's because we can use these arrangements combined with the morphology to come up with like staphylococcus or streptococcus. So streptococcus, like strep throat. Group A strep does cause strep throat. Staphylococcus, grape-like clusters, those are found on your skin. They can call methicillin-resistant Staphylococcus aureus. So these are important to look at for that type of thing. In terms of shapes and arrangements. Okay, we're going to have to pause here and erase. One moment. Boop. Okay, so I hope this is recording again. Let me see. Sure looks that way. Okay, so we talked about the bacterial shapes. We're also going to talk this week about the different bacterial features, some things that they all have, like cell membranes, nucleoids, ribosome, cytoplasm, we already know. What's interesting about bacteria is that they keep their DNA in a region of the cytoplasm called the nucleoid, and their DNA is like one chromosome, so it's like one circular chromosome always. They can, because this isn't in a nucleus, they can undergo transcription and translation at the same time, which is why bacteria replicate so much faster than eukaryotes. Some of them also have the ability to form endospores. These are not reproductive structures. These are like a survival bunker. They're not reproductive. And so the genus that do those that you should absolutely know, Bacillus species like Bacillus anthracis, it causes anthrax, and then Clostridium. Clostridium perfinigens, Clostridium causes gas gangrene. Clostridium difficile causes C. diff infections in the GI. Those endospores are really, really difficult to get rid of. You have to like oxidate the junk out of them or autoclave them or boil them for 6 hours or something really intense like that. So we'll talk more about those. And then we're also talking about the different features that maybe you haven't ever heard before that you should definitely kind of look up and familiarize yourself before class. The S layer, which is a protein layer, protective layer, the glycocalyx, which can be a slime layer, or the capsule. Some of these guys have outer membranes. Those are called gram negatives. Those outer membranes contain endotoxin, which causes sepsis. Cell walls. They all have cell walls. Some of them have peptidoglycan in their cell walls. A thick layer is gram-positive. A thin layer is gram-negative. Some of them have waxy cell walls. We see those in the Mycobacterium. Those are slow-growing. Those cause leprosy. Those cause tuberculosis. Some of them have an actin cytoskeleton that kind of helps support cell shape. Some of them form a capsule, which means that they are absolutely a pathogen. Some of them have inclusion bodies. Some of them have this plasmid, which is extra DNA. And some of these plasmids, like the R plasmid, is the resistance plasmid. These are like optional. They're not part of the main chromosome. Resistance plasmid causes antibiotic resistance, for example. Whereas the F plasmid causes them to form a pilus, which is a reproductive structure. Flagellum. Some of them have it, some of them don't. A lot of people think that flagellum is something that all of them have. Not true. Some of them are never even move. The archaeal features are very similar to this, but their flagella is more similar to a eukaryote because archaea are closer to eukaryotes. We're also going to talk about, of course, eukaryotic features. They're large, they have organelles, they're very fancy. We should absolutely know all the organelles and their functions, but, you know, that's kind of been beaten to death. Transport within the cell, how everything kind of starts at the nucleus, and this is evolutionary, that makes sense. Outside of the nucleus is the endoplasmic reticulum, that's like a big web and transport, and a lot of that, the rough ER is for making proteins, smooth ER is for making lipids. From that ER, they then get transported to the Golgi, which packages it to make vesicles. One of those vesicles is the lysosome, which contains digestive proteins and protects the cell. So we're also going to talk about the endosymbiotic theory. Which explains the origin of mitochondria and chloroplasts because, well, it's presumably, nobody was around then, we don't actually know for sure, but mitochondria and chloroplasts are the same size as bacterial cells. They also contain bacterial ribosomes, which are smaller. They're the 70S kind. Whereas eukaryotes have large ribosomes, they're 80S. So if you take an antibiotic, it messes up your mitochondria as well. They also have their own DNA. They also replicate independently. And they're really extremely interesting. So those are some of the ideas of endosymbiotic theory. They also have like an extra membrane compared to the other organelles, which might mean that they were brought into the cell through endocytosis, which coats whatever comes in with an extra piece of the membrane. Okay, we're also going to talk, for eukaryotes, we're also going to talk about the replication, which of course is mitosis and meiosis. We're also going to talk about so mitosis for regular cells, meiosis for sex cells. This results in haploid. These guys are diploid. We're also going to talk about schizogony, which is found in some protists. We're going to talk about budding, which is found in yeasts, where they end up with a teeny tiny copy of themselves. Super cute. And of course spores, which is again found in yeast. So these spores are reproductive, other ones are not. Oh, I keep running out of space on these guys. We're going to compare and contrast bacteria and archaea, the different types of protists, and then put that over here, different types of protists. Protists, of course, being the unicellular eukaryotes. They are so immensely diverse. Some of them are involved in disease, like Plasmodium causes malaria, trypanosoma, like bloodborne diseases, those types of things. So they are important to know, and your book classifies them by the different modes of locomotion, how they get around. So do they use fake feet, like amoebas? Like those are called pseudopods. Do they have cilia, which can be specialized structures only found in eukaryotes that can be used for feeding or also for movement? Do they have flagella? And of course, their flagella are huge and complicated. So that's like the general overview of everything that we're going to be talking about. Viruses, viroids, virions, and prions I talk about in a separate video. So I hope that's helpful. Oops.